Okay, so today's lecture is really brief. It is even briefer than yesterday's lecture because it's just a continuation of yesterday's lecture. Just a few interesting questions that you may have that are probably unanswered, definitely unanswered by my lectures, and another structural thing that may or may not appear on your practical. Oh my god, that was my hip. That just popped. Oh. Well, that'll make a good question. Pay attention to what I just said. Ow. Okay, so the sense continued. Physiology of vision, hearing, and equilibrium, although we're mostly talking about sight. Yeah. Your practical is tomorrow. It's Friday the 10th. Yeah. So hopefully you've been studying all week. If you haven't been, then it will be very hard for you to be successful, but you better start studying. You got to study all the structures we have gone over already, and you need to be familiar with the functions, the physiology of everything as well. If I didn't go over something, maybe, in a lecture, then you can definitely say that it won't be on the practical. All of it comes straight from my pre-recorded lectures, including this video. It'll be available from 9 a.m. to 9 p.m. Friday. And you'll have a minute per question. Right now, I think there's 40 questions, so it'd be 40 minutes long. And I'll be watching your screen. So do not leave Canvas online. Do not attempt to cheat. It would not be smart. I could definitely tell, judging by your answers too, if you think you're being slick by doing something else. So some of this may appear in your practical, so please pay attention. There will be participation questions from this presentation, and there will be a quiz on this presentation. So I have these two lovely stock photos. This is relevant information. Aren't they cute? So it's really three big concepts. You probably heard the term 20/20 vision before, or if not, at least listen to Justin Timberlake's album, The 20/20 Experience. And this was the year too, wasn't it? I remember at New Year's Eve, we were all saying, it's 2020. We're going to have 20/20 vision. This year is going to be it. But honey, little did we know. So what does it actually mean? 20/20, it's a ratio for visual acuity. And visual acuity is just a fancy way of seeing how well you can see something from a distance, how sharp or clear an image is. And what that ratio is saying is not that you have perfect vision, but actually you have average vision, being that you can see what an average human being sees from 20 feet away. So you'd be perfectly average. So that's a really big misconception, actually. But you know those eye charts that they make you read, I think it's something like at 9 millimeters, 20 feet away, if you can read it perfectly at 9 millimeters, you're average, or you have 20/20 vision. There is such a thing as 20/10 vision. There's such a thing as 20/40 vision, even 2200, which is the legal limit for being legally blind. A lot of people have 20/40, 20/30, 20/50, just slightly different ratios. That just means they have a little bit less than average sight. If you have 20/10 vision, it means you have very impressive vision. You're able to see things at 20 feet away that the average person can see 10 feet away. So you'd have very, very acute vision. And it's less common, but it's not impossible. There are people that have incredible sight, but there's no such thing as perfect vision. There's no such thing as perfect anything in science, really. It's all relative. So this particular schematic can be influenced by your genetics. You may have heard of astigmatism, nearsightedness, or farsightedness, and that's all influenced by genetics. That has to do with the shape of your eye. If it's bent a certain way, the lens won't focus the image correctly, or as accurately as it could otherwise. You can have worse vision depending on damage or stress to your eyes. Like if you stare at the sun too long, it can burn some of your photoreceptors and you'll have a harder time seeing. If you like to stay around really dark places, your vision can get a little wonky. Next concept, we're talking about the blind spot of the eye. I think I very briefly I don't think I talked about it. There's just a diagram that included the optic disc, so I'm putting a different one on here that's a little bit larger that you can see. So the optic disc is a physical structure within the very back of your eye, and it's also known as the blind spot. So it's actually the head of the optic nerve. So the nerve that actually carries the information from your eye to your brain to interpret it as sight and vision. The very head of it, the top of it, or rather the bottom, depending on how you're looking at it, is in the very back of the eye, and it is actually where all the ganglion cell axons exit the eye. So like the very beginning of that big thread of nerve. And they form the optic nerve, yes, after leaving the eye. The reason why this is called the blind spot is because your whole eye is lined with photoreceptors except for this area. It's just a bunch of nerves. There's no place for there to be any photoreceptors. So when light hits it, there's absolutely nothing going on there. You don't have a message from that one tiny spot on your eye. There is no photo detection. So you technically have a blind spot that you can't really focus on. And you can look up a fun way to test it online. There are different little activities you can do. But you can see it's right in the back of the eye, right there on that structure, against the flat of the back, going and turning into the actual optic nerve. So pay attention to the optic disc, and you might want to be able to tell me why you can't see anything from it. The last thing I'm going to go over is color blindness. Is another very interesting phenomenon that's actually pretty common in humans. I have a red-green colorblind picture up at the top, and then an example if you're not red-green colorblind. If you pass the test, you look down, you can see what it's like to be red-green colorblind. With this Fuji apple and this Granny Smith apple, you can see it just sort of looks yellowish because, well, I'll get to that. So colorblindness is typically inherited. And blindness isn't really a good term for it. A better word would be deficiency. The photoreceptors are failing to respond appropriately to the light that is hitting them. The variations in wavelengths of the light actually aren't perceived, or aren't perceived very well. Sometimes it has to do with the shape of the photoreceptor. Sometimes they're bent, sometimes they're absent. So cone deficiency or absence on the fovea, which is on the macula, these are structures that I went over in yesterday's lecture, is where the majority of your color interpretation comes from. And there are certain conditions that you can have that are mostly genetic, typically inherited, that will cause a either very small number of cones or a complete absence of cones on the fovea. And that just completely messes up your ability to distinguish between, I should say, red and green specifically, but also other colors, like shades of red, pinks, purples. Genetic cause, yeah. So, red-green color blindness is genetically linked. Other color blindness can come from, other types of color blindness can happen from diseases, damages to the eye, birth defects, or, etc. So why are males, I don't know if you know this, but males are much more likely to exhibit red-green color blindness. 8% of males compared to 1% of females have color vision problems in general, but when it comes to red-green color blindness, it is an overwhelming majority male. And the reason for this is that it's an X-linked recessive gene. If you can recall back to biology, you will know that the sex chromosome is, for males, an X and a Y, and for females, 2 X's. And when there's a sex-linked gene, it typically will always affect a male because they only have one X chromosome. And the X chromosome is one of the, not always, but usually the only chromosome that's fit to actually hold on to a trait, because the Y shape is actually smaller and missing. If you saw a picture, you would see that it's probably not efficient enough to carry a gene, but sometimes it does. But in this case, it doesn't. It's only X-linked. So if a male chromosome has the X-linked gene, it will show, because there's not another gene that's competing for it on a different X chromosome. Whereas with females, they have two X chromosomes, so if one is affected but the other isn't, there's likely a dominant trait that will override that from being expressed. However, they will still carry it. Females will only be affected if the mother is a carrier and the father is affected, while boys have a pretty good chance. If their father's affected, they're most likely going to have it. If their mother's a carrier, they might have it. It's a small chance, but it's a chance. And it's very common amongst males. Some of us probably have it, actually, in this class. And that's all I have for this slideshow. So just study this material. Don't ignore it because it's short. It is important, and you will have 2 more assignments on this, just like you had with the other ones.